13,000 feet below you in total darkness and crushing cold, the ocean floor is making oxygen. No sunlight, no photosynthesis. Just rocks, producing the air that life runs on. Scientists thought their equipment was broken. They spent 8 years trying to prove themselves wrong. They couldn't. We'll go down to the bottom of the Pacific, meet the creatures living in the dark, watch mining companies race to tear it all apart, and ask the question that is now shaking every biology textbook on Earth. If life-giving oxygen was already down there in the dark, where else might life be hiding? If this gets you, like and subscribe. Let's go. The year was 2013. A researcher named Andrew Sweetman was sitting aboard a research vessel in the middle of the Pacific Ocean, staring at a number on a screen that should not exist. His team had dropped sensors 13,000 feet down to the seafloor. That is deeper than the tallest mountain on land is tall. The sensors were supposed to measure how much oxygen sat at the bottom of the ocean. Standard stuff, routine work. They had done it dozens of times before. The reading came back. Oxygen levels were rising. Sweetman blinked. He looked again. Every scientist on that boat knew the same rule. In complete darkness, oxygen only disappears. Tiny organisms eat it. Chemical reactions consume it. Nothing down there makes it. Every textbook, every study, every experiment for 200 years agreed on that. Oxygen needs sunlight. Oxygen needs photosynthesis. Without light, you get nothing. His first thought was simple. The sensor was broken. Equipment fails at extreme depths all the time. The pressure down there is like stacking 700 cars on top of a single square inch. Electronics crack. Seals fail. Sensors drift. It happens. So the team recalibrated. They ran new tests. They swapped equipment. They adjusted the software. The reading stayed wrong, which is to say, it stayed exactly the same. Oxygen. Rising. In the dark. This was not a one-day problem. Sweetman's team kept returning to the same zone of the Pacific year after year. Each time, the same impossible result came back. They spent nearly 8 years assuming the fault was theirs. 8 years of chasing a ghost in their instruments. Picture that. 8 years of your career dedicated to proving that you made a mistake. 8 years of colleagues raising eyebrows. 8 years of publishing nothing because the data looked insane. Then, in 2021, they tried something different. They pulled in a completely separate measurement system, a backup method that works on an entirely different principle than the original sensors. If both systems agreed, the anomaly was real. If they disagreed, the original sensors were garbage. Both systems agreed. Sweetman described that moment as deeply unsettling. His team had just confirmed something that the entire history of ocean science said was impossible. Oxygen was being produced in a place where no known process could produce it, in total darkness, under crushing pressure. Miles beneath the surface. The question was no longer whether the readings were accurate. The question was what on earth was making the oxygen. And the answer, when they finally found it, was stranger than anyone on that boat had imagined. Down on the seafloor, scattered across the mud like forgotten rocks in a giant's garden, sat millions of lumpy, dark, potato-shaped formations. Nobody had paid them much attention before. They had been catalogued, photographed, filed away. But now, for the first time, scientists looked at them differently, because something about those rocks was very much alive in a way science had never considered. What the team discovered next would not just change oceanography, it would force scientists to rewrite the story of how oxygen arrived on Earth in the first place, and it would push astrobiologists to point their telescopes at places they had mostly ignored. The rocks at the bottom of the Pacific were doing something extraordinary, and the world was about to find out. For 200 years, the rule held firm. Oxygen comes from light. Period. It started with a Dutch scientist named Jan Ingenhousz in the 1700s. He noticed that plants submerged in water released tiny bubbles when sunlight hit them. In the dark, the bubbles stopped. He had just discovered photosynthesis, the process where plants use sunlight to convert carbon dioxide and water into sugar and oxygen. That single discovery became the backbone of biology. Every oxygen molecule in the air you are breathing right now came from light hitting a green leaf or a surface-dwelling algae. The Amazon rainforest, the grasslands, the oceans near the surface where sunlight still reaches, all of it powered by the same process Ingenhousz found in his little water tank centuries ago. And scientists extended that logic downward, deep into the ocean, where sunlight vanishes completely after about 600 feet. The math was simple. No light means no photosynthesis. No photosynthesis means no oxygen production. Deep ocean creatures survive on scraps of organic material drifting down from above, like snow falling through the water. They consume oxygen, they never make it. This became textbook law, unquestioned, repeated in every marine biology course on the planet. The deep ocean, in this model, is a place of pure consumption, a graveyard of energy. Nutrients go in, nothing new comes out. That model worked beautifully for 200 years because nobody had a reason to test it below a certain depth. The deep ocean is incredibly hard to reach. Sending sensors down 13,000 feet is expensive, time-consuming, and technically brutal. Most scientists studied the ocean's upper layers, where life is thick, light is present, and equipment survives easily. So the rule was never truly tested in the abyss. It was assumed. And assumptions have a way of surviving until someone stumbles onto the thing that breaks them. The first crack came not from a bold experiment designed to challenge the textbooks. It came from a routine survey, a team checking baseline oxygen levels at the seafloor. Data so expected to be boring that no scientific journal would have wanted to publish it. Except the data came back wrong. And as you already know, it stayed wrong for 8 years. Here is what makes this so significant for you personally. Every breath you take is tied to a story about where oxygen comes from. That story shapes how we look for life on other planets. It shapes what we think life needs to survive. It shapes the entire framework of biology from the smallest bacterium to the human brain. If that story has a missing chapter, the whole framework shifts. 200 years of certainty rested on the assumption that light is the engine of oxygen. Sweetman's data suggested something else was running in the background, something scientists had not accounted for, something that had possibly been running quietly for millions, maybe billions of years. The textbook rule was not wrong exactly. Photosynthesis is real, light does produce oxygen. That part stays true. The rule was incomplete. And incomplete rules in science are sometimes more dangerous than wrong ones, because nobody looks for what they already think they understand. Something on the seafloor had been producing oxygen the entire time scientists were looking elsewhere. And the clue to understanding it looked, to the untrained eye, exactly like a pile of dirty rocks. That pile was about to become the most important geological discovery of the decade. Close your eyes and picture the most lifeless place you can imagine. Now go deeper. The Clarion-Clipperton Zone sits at the bottom of the Pacific Ocean, halfway between Hawaii and Mexico. It stretches for 4,500 miles across the seafloor. To put that in perspective, that distance is wider than the entire continental United States from coast to coast. It is one of the largest underwater regions on Earth, and until recently, most people had never heard of it. Standing on the surface of the ocean above this zone, you would see nothing unusual. Blue water, waves, maybe a few distant clouds. The Pacific looks the same up there for thousands of miles in every direction. But 13,000 feet below you, the world is something else entirely. Sunlight disappears long before you get anywhere near the bottom. By 600 feet down, the ocean has filtered out almost all visible light. By 3,000 feet, you are in permanent, absolute darkness. The kind of dark where your eyes would never adjust, no matter how long you waited, because there is zero light to adjust to. At 13,000 feet, the pressure is crushing. Every square inch of your body would experience a force equivalent to stacking about 50 cars on top of it. No human has ever stood on the Clarion-Clipperton Zone floor. Our bodies would collapse instantly. The temperature hovers just above freezing, a few degrees warmer, and the water would start to circulate differently. A few degrees colder and it would ice over. Life here sits at the absolute edge of what physical chemistry allows. And yet, the floor is not empty. When robotic cameras sweep across the Clarion-Clipperton Zone, they show something almost serene, a flat, muddy plain stretching in every direction. The sediment is fine and pale, disturbed by almost nothing. There are no currents strong enough to ripple it. There are no storms down here. The seafloor in this zone has been sitting almost completely still for millions of years. And scattered across that pale mud, as far as the camera can see, are the rocks. Dark, lumpy, roughly the size and shape of russet potatoes. Millions of them spread unevenly across the mud like someone scattered them by hand from a great height. They sit alone, in pairs, in clusters. Some are half buried. Some sit completely exposed on the surface. They do not move. They barely change. They have been sitting on that mud for an almost incomprehensible length of time. These are polymetallic nodules, and for a long time scientists looked at them the same way you would look at a pile of rocks on a sidewalk. Interesting, worth cataloguing, nothing more. The zone was first identified as scientifically significant in the 1970s, mostly because researchers realized the nodules contained valuable metals. Mining companies started paying attention soon after. But the nodules themselves, as objects of deep scientific curiosity, did not get serious study until recently, because nobody asked what the rocks were doing. Now that someone finally did, the answer changed everything. Those dark, potato-shaped lumps, sitting motionless on the floor of the Pacific, were doing something that required rewriting the rules of biology, reconsidering the origin of life, and potentially redirecting the search for alien worlds. And the deeper you go into what they are made of, the stranger the story gets. Imagine standing at the bottom of a swimming pool. You can feel the pressure in your ears, a slight squeeze, a little discomfort. That is maybe 3 feet of water above you. Now imagine 13,000 feet of ocean stacked on top of you instead. The pressure at the depth of the Clarion-Clipperton Zone is roughly 390 times greater than what you feel standing on land. Scientists call this unit of measurement 1 atmosphere at sea level. At 13,000 feet, you are sitting under about 390 atmospheres. If you compressed a sealed basketball to that pressure, it would shrink to a fraction of its original size and stay that way. Human lungs would collapse in less than a second. Steel bends, glass cracks, even specially designed scientific equipment sometimes fails under that kind of force. This pressure shapes everything about how life works down there. Creatures living in the abyss have had to evolve entirely different body chemistry. Their proteins are structured differently than ours. Their cell membranes are more flexible. Their enzymes work under conditions that would instantly destroy the same molecules in a surface organism. Some deep-sea fish have no swim bladder at all because the pressure would crush any gas-filled cavity. Others produce strange chemical compounds that essentially act as internal pressure buffers, molecules that stop their cells from being crushed by the weight of the ocean above. And here is what makes the dark oxygen discovery even more remarkable. Whatever process the polymetallic nodules are running down there, it works under those 390 atmospheres of pressure. That is not a detail to pass over quickly. Chemical reactions behave differently under extreme pressure. Some reactions speed up, others slow to almost nothing. Some compounds that are stable at sea level become reactive, some that are reactive become inert. The whole chemistry of the deep ocean plays by subtly different rules than the surface world. When Sweetman's team first tried to figure out how the nodules might be generating electricity, pressure was one of the first complications they ran into. Replicating those exact conditions in a laboratory required specialized chambers that only a handful of research institutions on Earth even own. The team eventually built a pressure simulation in a lab. They chilled the water to near freezing. They compressed it to match the conditions of the Clarion-Clipperton zone. Then they watched what happened when they placed clusters of nodules together inside the chamber. The oxygen appeared, confirmed under controlled conditions, in 2024. Not a sensor glitch. The nodules, under pressure, in cold and total darkness, produced oxygen. The pressure, rather than shutting down the process, seemed to be part of what made it work. The deep ocean was not fighting the chemistry, it was enabling it. That understanding cracked open a much bigger question. If this works under conditions we once called hostile to chemistry, then maybe our definition of hostile is wrong. Maybe the environments we have been calling dead zones are actually engines. And if that is true in the Pacific, the implications reach far beyond Earth, because there are other places in our solar system with crushing pressure, total darkness, and liquid water. You are about to find out exactly where they are and why they matter more than most people realize. Picture being told you are wrong every single day. For 8 years, that is what Andrew Sweetman's team lived through. And the strangest part is, they were the ones telling it to themselves. After the first set of impossible readings came back in 2013, the team did what any good scientist would do. They assumed human error. They checked the calibration on every sensor they had used. Calibration is the process of making sure your measuring device is accurate, basically tuning it before use. They found nothing wrong. The sensors read correctly. So they went back to the seafloor. They ran the measurements again. Oxygen rising. In the dark. Again. They swapped the sensors for entirely different units from a different manufacturer. Same result. They adjusted the way they were deploying the equipment, changing the angle, the speed of descent, the way it sat on the mud. Same result. They questioned whether some contamination from the ship above might be reaching the sensors and producing a false reading. They redesigned their deployment cables. Same result. Year after year, the same number came back. Oxygen production, in a place where oxygen production was scientifically illegal. The team published almost nothing about it during those years, because in science you do not publish data you cannot explain and cannot confirm. You would be laughed out of the field. Without a mechanism, without a repeatable experiment, without a working theory for how the oxygen was appearing, the data was unpublishable. So they sat on it. They worked quietly. They kept going back. In 2016, a different team of researchers working in a completely separate part of the Pacific reported similar anomalies. Oxygen levels in deep water where oxygen should only be disappearing. That news reached Sweetman's group and had a double effect. On one hand, relief. Someone else had seen it too. On the other hand, pressure. Because now the anomaly was repeating in multiple locations and the scientific community was going to start asking questions. By 2019, Sweetman's team had narrowed the possible explanations down to 3. A biological source, meaning some unknown organism was producing the oxygen. A chemical source, meaning some reaction between seawater and the seafloor minerals was doing it. Or an electrical source, meaning some form of natural current was splitting water molecules apart. The biological option seemed unlikely. No known organism at that depth produced oxygen at those rates. The chemical option was possible, but did not fully explain the voltage readings they had started picking up near the nodules. That left the electrical source as the leading theory. But a theory needs proof, and proof at 13,000 feet requires equipment that most universities in the world cannot afford. The team spent 2 more years securing funding, building new tools, and designing the backup measurement system that would finally settle the question. In 2021, they went back down one more time. And this time, the backup system agreed with everything the original sensors had been saying since 2013. 8 years of doubt erased in a single data run. The machine had never been broken. The planet had been doing something they were not prepared to see, and once they accepted that, they finally had to ask the question they had been avoiding. What, exactly, was doing this? By 2021, Andrew Sweetman's team had run out of ways to blame themselves. They had swapped sensors, redesigned deployment systems, eliminated contamination, ruled out biological sources, spent 8 years building the case that their equipment was fine, and the data was simply impossible. And impossible data, in science, is the most dangerous kind, because it either means you made a mistake, or it means reality is stranger than your models. The team decided to find out which one it was. The backup measurement system they used in 2021 works on a completely different principle than a standard oxygen sensor. Regular sensors detect oxygen molecules directly, chemically. The backup system they deployed measures oxygen indirectly, using something called optode technology. An optode works by shining a tiny light onto a special chemical dye. Oxygen in the water changes how that dye glows. The device reads the glow and calculates the oxygen concentration. Two completely different technologies, two completely different physical principles. If both agree, the reading is real. They lowered the new system to 13,000 feet. They let it sit on the sea floor beside the nodules for the same amount of time they had always used. They waited. The optode returned the same numbers as the original sensors. Oxygen. Rising. In the dark. Sweetman later said the confirmation felt deeply unsettling rather than exciting, because excitement assumes you understand what you found. His team did not understand it yet. They had confirmed that something impossible was happening without knowing why. But the confirmation unlocked the next step. With real data they could now defend publicly, the team started running physical samples. They brought nodules up from the seafloor and into a specially built pressure chamber on their research vessel. Inside the chamber, they recreated the conditions of the Clarion-Clipperton Zone as closely as possible: near freezing temperature, high pressure, total darkness, seawater drawn from the same depth. Then they placed clusters of nodules together and measured what happened. Oxygen appeared in the water. This was the moment that shifted everything. The nodules were not just sitting passively on the seafloor, they were actively doing something to the water around them. The team measured the voltage coming off individual nodules and found readings up to 0.95 volts per nodule. That number matters for a reason that becomes clear very quickly. To split a water molecule into hydrogen and oxygen, you need a voltage of 1.5 volts. One nodule alone cannot do it. But when several nodules cluster together in the mud, their voltages add up. Cluster enough of them, and you cross the 1.5 volt threshold. The seawater around the cluster splits. Hydrogen goes one way, oxygen goes another. The oxygen dissolves into the water and starts rising slowly through 13,000 feet of ocean. That process has a name: electrolysis, the same reaction used in high school chemistry demonstrations, happening naturally, in the dark, on the floor of the Pacific Ocean. The team brought their data to peer review and published their findings in July 2024 in Nature Geoscience, one of the most respected scientific journals on Earth. The paper confirmed dark oxygen production as a real, documented phenomenon. And then the phone calls started coming in from places the ocean science community had not expected. From NASA. For a long time, they were just rocks. That is the honest summary of how the scientific community treated polymetallic nodules for most of the 20th century. They were catalogued, photographed, measured. A few researchers wrote descriptive papers about their shape and distribution, and then the files went onto shelves, and the nodules went back to sitting on the seafloor doing what rocks do. They got slightly more attention in the 1970s, when ocean surveys discovered that the nodules were incredibly rich in valuable metals. Mining companies sent their own research vessels to map where the largest deposits were. The Clarion-Clipperton Zone, with its enormous field of nodules spread across mud wider than the United States, became one of the most commercially appealing underwater regions on Earth. But even the mining industry was interested in what the rocks contained. Nobody was particularly curious about what the rocks were doing. To understand why the nodules ended up being so significant, you need a picture of what they actually are. A polymetallic nodule starts as almost nothing. A tiny grain of sediment, sometimes a shark tooth, sometimes a microscopic piece of volcanic rock. That seed particle lands on the seafloor and sits there. Over millions of years, minerals dissolved in the seawater above slowly precipitate out and coat the particle. Layer by layer, the way an onion grows its rings, the nodule builds itself outward. The growth rate is almost impossible to comprehend. Scientists estimate that a single nodule grows about 1 to 2 millimeters every million years. Your fingernails grow faster in a single day than a nodule grows in a million years. A nodule the size of a potato is likely tens of millions of years old. The minerals deposited layer by layer include manganese, iron, nickel, cobalt, and copper. These are dense, heavy metals that accumulate in those cold, dark, high-pressure conditions because the chemistry of deep seawater keeps them in solution until they find a surface to cling to. Cut a nodule open and you can see its history. Each ring represents a different geological era, a different chemical environment, a record of the ocean's chemistry stretching back tens of millions of years, written in concentric circles of dark mineral deposits. Now here is the thing that nobody accounted for until very recently. Those metal layers are not just passive deposits. Manganese and certain other metals can act as semiconductors. That means they can move electrical charge under the right conditions. The layered structure of the nodule, different metals stacked in different concentrations, creates something that functions like a natural battery. Opposite charges build up on different layers. A voltage difference appears across the nodule's surface. Scientists did not design this. The ocean built it over millions of years, one microscopic layer at a time. And now, sitting in clusters on the seafloor, millions of these natural batteries were quietly doing what batteries do, generating electricity. The question of why they hold that charge and what exactly maintains it over geological time remains a contested and open area of science. The mechanism is real and confirmed. The full explanation is still being worked out. But the next discovery was even more immediate, because those voltages were doing something very specific to the water around the nodules. Something that should have been impossible. Nobody set out to measure the electrical charge on a deep-sea rock. It happened almost by accident. During the seafloor surveys that Sweetman's team was running after 2021, one of the instruments deployed alongside the oxygen sensors was a simple voltage probe, the kind of device that measures electrical potential in a medium, the same basic technology as a multimeter you would find in a hardware store, just built to survive 13,000 feet of ocean pressure. The probe was there to help understand the chemical environment around the nodules. Voltage gradients in seawater can indicate all kinds of things about local chemistry. It was background data. Context for the oxygen readings. Then the probe logged something unexpected. The nodules were carrying voltage. Individual nodules, sitting in the cold, dark mud, were generating a measurable electrical potential on their surface. The readings varied depending on the size and mineral composition of each nodule, but a significant number of them registered close to 1 volt. For context, a standard AA battery that powers your TV remote produces 1.5 volts. These rocks on the seafloor were producing roughly 2/3 of that, sitting completely alone in the dark, with no external power source anyone could identify. The team tested nodule after nodule. The voltage was consistent. It was not a spike caused by some passing current in the water. It was steady, persistent. The nodules carried their charge the way a capacitor holds energy, slowly built up and slowly released. The next logical question was immediate. Where was the charge coming from? The leading theory, which is supported by laboratory work but not yet fully confirmed, is that the layered mineral structure of the nodule acts like a natural electrochemical cell. When different metals sit in close contact within a conductive fluid like seawater, they create a potential difference between them. The manganese-rich outer layers interact with the iron-rich inner layers, and the seawater surrounding the nodule completes the circuit. The result is a voltage, slow, steady, and persistent. This is the same basic principle behind every battery humans have ever built. A battery is two different materials with different chemical properties, sitting next to each other in a conductive fluid, producing a voltage because the materials want to exchange electrons. We call that chemistry. The ocean figured it out millions of years earlier using manganese and iron instead of lithium or zinc. The voltage on a single nodule is real, confirmed, measured multiple times. The origin of that voltage is a working theory with strong experimental support, but the full mechanism is still actively researched. And here is where it gets stranger. Nodules on the seafloor do not sit perfectly spaced apart like items on a shelf. They cluster, they pile near each other, they touch, they sit in contact with the same conductive sediment and seawater. And when multiple voltage sources connect in sequence, their voltages add together. 10 nodules clustered in the mud, each producing close to 1 volt, can collectively generate far more than any one of them could alone. Enough, as it turns out, to cross a very specific and very important threshold. And crossing that threshold was doing something to the water that the whole scientific world was about to have to explain. Water looks simple. 2 hydrogen atoms, 1 oxygen atom, locked together in a tight molecular bond. Breaking that bond is harder than it sounds. To force a water molecule apart, you need to push enough electrical energy into it to overcome the force holding the hydrogen and oxygen together. Scientists have measured exactly how much energy that requires. The threshold is 1.5 volts. Apply less than that, and the water molecule stays intact. Apply 1.5 volts or more, and the molecule splits. Hydrogen goes to the other. This process is called electrolysis. Humans have been using it industrially for over 100 years to produce hydrogen fuel, to purify metals, and to generate oxygen in sealed environments like submarines and space stations. It requires a power source, a battery, a generator, some deliberate input of electrical energy from outside the system. The deep ocean had none of those things. Or so everyone thought. Here is what the Clarion-Clipperton Zone actually looks like at the nodule scale. The seafloor is not covered in a perfectly even layer of rocks. The nodules sit in clusters, touching each other, partially buried in the same conductive mud, surrounded by the same seawater. That seawater is a conductor. The mud is a conductor. The nodules themselves carry their individual voltages. When you connect multiple voltage sources through a conductor, the voltages add up. This is basic electrical physics, the same principle behind connecting batteries in a series to make a flashlight brighter. A single nodule sitting alone in the mud might generate 0.95 volts. Below the threshold, nothing happens to the water around it. But put 3 nodules in contact. 4. Their voltages combine through the conductive seawater and sediment. When the total voltage in that cluster crosses 1.5 volts, the seawater touching those nodules begins to split. Hydrogen bubbles form. Oxygen molecules dissolve into the water. The oxygen does not stay near the seafloor. It is lighter than the surrounding seawater, so it begins a slow drift upward through 13,000 feet of ocean, mixing into the water column above. Sweetman's team confirmed this in their pressure chamber experiment. They took nodule clusters from the Clarion-Clipperton Zone, placed them in chilled seawater under high pressure, and measured what happened. Oxygen appeared, consistently, reproducibly. The amount of oxygen produced correlated with the combined voltage of the cluster. This is now confirmed science, published in Nature Geoscience in 2024. The Clarion-Clipperton Zone alone covers an area wider than the continental United States. Across that entire region, millions of nodule clusters sit in contact with each other, And with conductive sediment, every cluster large enough to cross the voltage threshold is quietly splitting seawater in the dark. Scientists do not yet know the total scale of dark oxygen production across the global ocean. That number remains a genuine unknown. But the mechanism is real, repeatable, and running constantly at the bottom of the Pacific. And those nodules, by doing nothing more than sitting in clusters in the cold mud the way they have for millions of years, were producing oxygen long before any human ever breathed. Which raises a question that changes everything. What if life on Earth found that oxygen first? Walk into any high school chemistry class and you will probably find a demonstration involving a battery, 2 wires, and a glass of water. The teacher connects the wires to the battery and drops the ends into the water. Within seconds, tiny bubbles appear on both wires. One wire produces hydrogen, the other produces oxygen. The water is splitting apart in real time, driven by the electrical current from the battery. The teacher calls it electrolysis. The students write it down. The class moves on. What nobody in that classroom realizes is that the same reaction, with no battery and no teacher and no wires, has been running at the bottom of the Pacific Ocean for millions of years. The principle is identical. Apply enough voltage to water and the molecules break apart. Hydrogen and oxygen separate. The difference at the seafloor is that the voltage does not come from a battery purchased at a hardware store. It comes from the electrochemical properties of minerals that the ocean has been layering onto rock seeds for tens of millions of years. Nature reinvented electrolysis before humans understood chemistry well enough to name it. That comparison matters because it makes the process approachable. Electrolysis is not exotic. It is not mysterious. We use it in industry every single day. Water treatment plants use it. Hydrogen fuel production uses. Oxygen generation systems on submarines and the International Space Station use it. It is one of the most well-understood electrochemical reactions in science. The discovery at the Clarion-Clipperton Zone does not challenge the chemistry of electrolysis. The reaction works exactly the way chemistry predicts it should. What it challenges is the assumption that conditions for electrolysis could only exist where humans deliberately set them up. The ocean created the conditions on its own, over geological time, through nothing more than the slow layering of minerals in cold seawater. And because the process is so well understood, scientists had a significant advantage in studying it. They already knew how to measure it. They already knew what to look for. Once Sweetman's team confirmed the voltage on the nodules and the oxygen in the water, the connection to electrolysis was almost immediate. The hard part was not identifying the process. The hard part was accepting that the process was happening at all. Human assumptions about where electrolysis can run were built entirely around human-made systems. A power source, a controlled environment. The idea that the seafloor could organize the same reaction on its own, with no human input, across thousands of miles of mud, was outside the mental model most scientists were working with. The discovery forced a specific and uncomfortable question. If this process runs naturally in the ocean, where else in the universe might it be running? What other environments, too dark or too cold or too hostile seeming for life as we know it, might actually be producing the building blocks that life needs? The answer to that question started not in the Pacific Ocean. It started with the rocks themselves and what is actually packed inside them. Because those same metals doing electrolysis at the bottom of the sea are also sitting in the device you carry in your pocket every single day. Pull out your phone. Look at it. The battery inside that device contains cobalt. It contains nickel. It contains manganese. Those three metals combined in specific ratios hold the electrical charge that powers everything your phone does. Every text message, every video, every notification. Your entire digital life depends on those three metals being present in the right proportions in a compact cell you can hold in one hand. Now picture 13,000 feet below the Pacific Ocean, the mud at the bottom of the Clarion-Clipperton Zone, the dark lumpy nodules scattered across it, Those nodules contain cobalt, they contain nickel, they contain manganese, the same metals in remarkably similar proportions to what modern battery manufacturers look for. Nature, over tens of millions of years, built battery-grade mineral deposits on the seafloor. And nobody noticed until mining companies started calculating how much money was sitting down there in the mud. The concentration of metals in polymetallic nodules is exceptional by any geological standard. Mining ore from the land surface typically yields a small percentage of target metal per ton of rock extracted. The nodules from the Clarion-Clipperton Zone yield dramatically higher concentrations per unit of material. This is why 17 separate mining contractors have already secured exploration licenses for the zone. The global demand for these metals is accelerating fast. Electric vehicles require large battery packs built from the same metals inside nodules. Renewable energy storage systems use them. As the world tries to move away from fossil fuels, the demand for battery metals is projected to rise sharply over the coming decades. The seafloor looks from a pure resource perspective like the answer to a supply problem that governments and corporations are increasingly worried about. But here is what the dark oxygen discovery added to that calculation. Those nodule clusters are producing oxygen. They are supporting ecosystems. They are possibly doing something that life on Earth has depended on in ways scientists are only beginning to understand. The metals inside the nodules are not inert rocks waiting to be collected. They are active participants in deep ocean chemistry. They maintain electrical charges. They enable a process that splits water and generates oxygen. They are part of a system that took millions of years to build. Extracting them would be fast. A commercial mining operation in the Clarion-Clipperton Zone could remove nodules at an industrial rate. The nodules grow back at 2 millimeters per million years. That gap between extraction speed and recovery speed is one of the central tensions the dark oxygen discovery dropped into an already complicated argument about deep-sea mining. The metals are valuable. The process they enable is remarkable. The question of which matters more, the economic value sitting in the nodule or the biological and chemical role the nodule plays on the seafloor, is a question with enormous consequences. And underneath all of that is a stranger and more fundamental question. How long have those nodules been running this process? And has life been taking advantage of it all along? A nodule the size of a baseball took longer to form than humans have existed as a species. Sit with that for a moment. Modern humans have been on Earth for roughly 300,000 years. The nodule sitting on the seafloor in the Clarion-Clipperton Zone started growing long before our ancestors stood upright. It was already ancient when the dinosaurs disappeared 66 million years ago. Some of the largest nodules in the zone began forming before mammals became the dominant life form on land. The growth process is almost impossibly slow, 1 to 2 millimeters of mineral deposit per million years. That number is confirmed by radiometric dating of nodule layers, a process that measures the decay of radioactive elements inside each mineral ring to calculate when that layer was deposited. Each layer records something. The mineral composition of each ring reflects the chemistry of the ocean at the time it was deposited. A ring from 50 million years ago contains the chemical signature of a 50 million year old ocean. A ring from 5 million years ago reflects the chemistry after major shifts in Earth's climate. Scientists can read the nodules' rings the same way a geologist reads rock strata, as a timeline of changing conditions. That means these rocks are geological archives. They hold a record of ocean chemistry stretching back tens of millions of years. Inside a single potato-sized nodule, you can find a compressed history of the Pacific Ocean that no other single object on Earth can provide. And here is why that matters for the dark oxygen story. If the nodules have been generating voltage and producing oxygen for tens of millions of years, then dark oxygen production is not a new process. It is not something that just appeared recently. It has been running continuously, or something close to continuously, for a stretch of time that makes all of human history look like a single breath. That changes the scale of what scientists are dealing with. Dark oxygen production, across the full extent of the Clarion-Clipperton Zone, and potentially across other nodule fields in other ocean basins, would represent a source of oxygen that has been contributing to deep ocean chemistry for geological timescales. Organisms living in the deep sea over those millions of years would have had access to that oxygen. Evolutionary pressures in the deep abyss may have been shaped by the presence of this oxygen in ways no scientists previously accounted for. The origin of certain deep-sea life forms may look different now. Species that biologists assumed were adapted to an entirely oxygen-poor environment may have actually evolved in the presence of a steady, dark-sourced oxygen supply provided by the nodules around them. That is a significant shift in the evolutionary story of deep ocean life, and it is one scientists are only beginning to reconstruct, because now they need to figure out exactly how much oxygen the nodules have been producing and for exactly how long. Those numbers remain unknown, but finding them will force a rewrite of a chapter in Earth's biological history that most scientists thought was already finished. The story was far from finished, and the next discovery made that clearer than anything. Scientists called the deep seafloor a desert. They were wrong. For decades, the standard image of the Clarion-Clipperton Zone's seafloor was a lifeless mudplain. The reasoning made sense at the time. No sunlight means no photosynthesis. No photosynthesis means no plant life. No plant life means nothing for animals to eat. The assumption was that anything living down there survived on a thin trickle of organic material drifting down from the surface, like crumbs falling from a table 13,000 feet above. Under that model, the deep seafloor should support only minimal, sparse, barely surviving life. Maybe a few worms, some microbial mats, nothing thriving. When researchers started collecting biological samples from the Clarion-Clipperton Zone during surveys, over the past decade, the results contradicted that picture completely. The zone contains one of the most diverse deep-sea ecosystems ever documented. Scientists have catalogued more than 5,000 species from this single region. The majority of those species had never been named before. Many of them had never been seen by any human, ever, in any ocean on Earth. The variety is startling. There are sponges that filter particles from the water with structures finer than silk. There are worms that live buried in the mineral-rich sediment around the nodules. There are sea cucumbers moving slowly across the mud, processing the sediment the way earthworms process soil on land. There are starfish, brittle stars, and mollusks adapted to a life in permanent darkness and near-freezing water. And clustered around the nodules specifically, researchers kept finding life in higher densities than in the flat mud between clusters. The nodules themselves serve as hard surfaces in an otherwise soft mud environment, and hard surfaces in the deep ocean are prime real estate. They offer attachment points for organisms that need solid ground to settle on. But the oxygen connection makes the nodule-centered life cluster even more significant. The dark oxygen production from nodule clusters adds dissolved oxygen to the immediate water around them. That local oxygen supply, even if small by surface ocean standards, may be enough to support aerobic organisms, creatures that need oxygen to survive, in zones of the seafloor where the background oxygen level would otherwise be too low. The nodules may be creating microhabitats, small pockets of relatively oxygen-rich water on an otherwise oxygen-starved seafloor. Life clusters around them the way hikers cluster around a water source in a desert. Confirmed, the ecosystem exists. The diversity is documented. The connection between nodule clusters and higher local biodiversity is observed and studied. The exact role dark oxygen plays in sustaining this ecosystem remains an active research question. But the correlation between nodule density and species density is consistent enough that researchers take it seriously. And among the creatures living in this hidden world, one caught the attention of the public more than any other, because it looks like something out of a dream. And it glows. In 2016, a remote-controlled camera drifting above the seafloor of the Pacific Ocean captured something nobody had seen before. a small pale octopus, completely white, almost translucent, moving with slow, delicate arm movements across the surface of a polymetallic nodule at 4,000 meters down. Scientists nicknamed it the Casper octopus after the Friendly Ghost cartoon because of its ghostly white color. The animal lacks the pigmentation that other octopuses use for camouflage, likely because in total darkness, camouflage is irrelevant. There is nothing to hide from that can see you. The Casper octopus was later confirmed as a new species, and its behavior turned out to be directly tied to the nodules. Female Casper octopuses lay their eggs attached to the stalks of dead sponges that grow on the nodule surfaces. They then wrap themselves around the egg cluster and guard it, sometimes for months without eating. The sponges that host those eggs grow on the nodules. The nodules are essential to the octopus completing its reproductive cycle. Remove the nodules, and the sponges lose their substrate. The sponges die. The octopuses lose their nesting sites. The species has nowhere left to reproduce. This direct dependency is confirmed and documented by researchers from the Natural History Museum in London and other institutions. The Casper octopus is one example, but the deep zone is full of creatures with similar dependencies. Many of the 5,000-plus species found in the Clarion-Clipperton zone are attached to nodules, live inside nodule clusters, or depend on the specific chemical environment around them. Some of the creatures in this zone produce their own light through a process called bioluminescence. They carry molecules in their tissues that react with oxygen to produce a cold glow. No heat, no fire, just chemistry producing light in the dark. Certain worms found in the zone flash blue-green light when disturbed. Some jellyfish species trailing through the water column above the zone glow in patterns that scientists believe are used for communication or predator deterrence. The bioluminescence is driven by oxygen, the same oxygen the nodules may be helping supply to the local environment. Take away the nodules, and you interrupt a chain that reaches from the seafloor mud through the ecosystems built on top of it, through the creatures reproducing on the nodules, through the organisms sustained by the chemical environment the nodules create, all the way up to the ghost white octopus guarding her eggs in the dark. That chain took millions of years to build. It cannot be rebuilt in any human time frame. And somewhere above this fragile, glowing world, 17 companies are preparing to harvest the rocks it all depends on. But before we get to that collision, the dark oxygen discovery opened up, one that reaches much further back in time than mining contracts, one that touches the very beginning of life on Earth. Every biology class teaches the same story. Life started in the sunlit ocean near the surface. Simple photosynthetic organisms evolved first. They started producing oxygen. Over hundreds of millions of years, oxygen built up in the atmosphere. That oxygen allowed more complex life forms to evolve. Eventually, animals capable of breathing oxygen appeared. The story has a clean logic. Light in, oxygen out, life follows. The dark oxygen discovery does not erase that story, but it punches a significant hole in the assumption underneath it. The assumption is this: oxygen was absent from the deep ocean until surface photosynthesis produced enough to slowly mix downward. Under that model, complex life dependent on oxygen could only evolve near the surface, where the oxygen was being made, and slowly spread as oxygen diffused into deeper water over geological time. If polymetallic nodules have been producing oxygen in the deep ocean for tens of millions of years, that assumption changes. The deep ocean may have had a local oxygen supply long before surface photosynthesis could have reached those depths. Aerobic organisms, life forms that run on oxygen, may have had conditions available to them in the deep ocean that scientists previously assumed were impossible down there. This opens a specific and genuinely contested question in biology. Could complex aerobic life have originated in the deep ocean, near the nodules, rather than near the surface? This is classified as a theory with supporting evidence, not a confirmed fact. The evidence is the nodule oxygen production mechanism. which is confirmed. The evolutionary implications are debated actively among researchers. Here is what makes it compelling. The deep ocean offers some unusual advantages as a birthplace for life. The temperature is stable. The chemistry is consistent over millions of years. The pressure is extreme, but pressure itself does not destroy chemistry. It changes it. And the nodule clusters create microenvironments with locally elevated oxygen, minerals, and electrical voltage. All three of those things, oxygen, minerals, and electrical energy, are considered essential ingredients in origin of life research. Some scientists studying the origin of life focus on hydrothermal vents on the seafloor as a possible cradle of early life. The dark oxygen discovery adds the nodule fields as another candidate environment. Different mechanism, similar concept. Deep ocean chemistry producing the conditions life needs. The scientific community has not reached consensus on this. The debate is active, serious, and involves researchers from biology, geology, chemistry, and planetary science all pulling in slightly different directions. What everyone agrees on is that the discovery forces a revision. The origin of life story needs a new chapter. The clean model, where oxygen only came from the surface and only spread downward over time, is no longer sufficient to explain what the seafloor is actually doing. And once scientists started pulling on that thread, they looked up from the Pacific Ocean floor and out toward the rest of the solar system. Because if oxygen can appear in a cold, dark, high-pressure ocean with no sunlight, then the universe just got a lot more interesting. For most of the 20th century, origin of life research focused on warm, shallow water. The classic image was a warm little pond, sunlit and chemical-rich, where the first self-replicating molecules came together, or a shallow coastal sea warm enough to encourage chemistry, close enough to the surface for UV light to drive reactions. The deep ocean was considered a dead end for this line of research. Too cold, too dark, too far from the energy sources life seemed to need. That view started cracking in 1977 when scientists discovered hydrothermal vents on the seafloor near the Galapagos Islands. These vents belch superheated water rich in minerals and chemicals from cracks in the ocean floor. Around the vents, entire ecosystems thrived with no connection to sunlight whatsoever. Tube worms, 6 feet long, crabs, clams, all of it powered by chemistry, not light. Those vent communities forced the first serious revision to origin of life thinking. Life, it turned out, could run on chemical energy instead of sunlight. The deep ocean was not a dead zone. It was an alternate engine. The dark oxygen discovery pushes that revision further. Hydrothermal vents are powerful but local. They occur at specific geological sites where the ocean floor is cracked and heated from below. They are dramatic and chemically intense, but they cover a tiny fraction of the seafloor. The polymetallic nodule fields of the Clarion-Clipperton Zone cover an area wider than the continental United States. If nodule fields like this exist in other ocean basins, and early evidence suggests they do, then the potential habitat for dark oxygen production is not a few isolated spots on the seafloor. It is a vast, distributed network spanning multiple ocean regions. A distributed, stable, low-energy oxygen supply across thousands of miles of seafloor is a very different environment from a violent local hydrothermal vent. It offers more gradual chemistry, more consistent conditions, more time for slow processes to accumulate complexity. Some researchers now argue that the nodule environment could have been a stable incubator for early aerobic life over a timescale that far exceeds anything possible near a vent. This is a theory. It is supported by the confirmed mechanism of dark oxygen production, by the geological age of the nodule fields, and by laboratory studies of how early life chemistry works under high pressure. It is contested by researchers who favor other origin of life models, and who point out that confirming any specific origin of life hypothesis requires evidence that is almost impossible to retrieve from billions of years ago. What is confirmed is this: the conditions needed for aerobic life exist in nodule fields, oxygen, mineral-rich surfaces, electrical voltage, stable temperature over millions of years. Whether life took advantage of those conditions at the very beginning is a question science cannot yet answer definitively. But the fact that the question can now be asked seriously, with real evidence underneath it, represents a genuine shift in how biology looks at its own history. And when scientists looked at other places in the solar system through that new lens, one moon kept coming up in conversation. A moon covered in ice, with a dark ocean underneath. For decades, the search for life in the solar system followed one guiding principle: follow the water. Water is the universal solvent that life as we know it depends on. Every living thing on Earth needs it. So scientists pointed their instruments at places where water might exist. Mars, which once had liquid water on its surface. Venus, whose early history may have included oceans. The icy moons of the outer solar system, where liquid water might hide beneath frozen surfaces. But following water was always paired with a second assumption. The water needed to be warm enough, close enough to a star, or near enough to a heat source, for chemistry to run fast enough to produce life. Cold, dark water seemed like a dead end, too slow, too chemical poor, too far from the energy that life needs. The dark oxygen discovery breaks that second assumption. The Clarion-Clipperton zone is cold. It is dark, it is under crushing pressure, and it is producing oxygen through a process that requires no heat and no sunlight. The energy comes from the minerals themselves, from the electrochemical properties of metals deposited by slow geological processes over millions of years. If that can happen in the Pacific Ocean, it can happen anywhere a similar set of conditions exists. Cold water, conductive minerals, time, and astrobiologists know exactly where those conditions might exist elsewhere in the solar system. Europa is a moon of Jupiter roughly the size of Earth's moon. Its surface is a cracked sheet of ice. Underneath that ice, a liquid ocean is kept fluid by the gravitational pull of Jupiter kneading the moon's interior and generating heat through a process called tidal flexing. That ocean is in total darkness. It sits at crushing pressures. Its floor is covered in rock. Enceladus is a small moon of Saturn. It is even more remarkable than Europa in one specific way. Enceladus actively shoots water into space through geysers at its south pole. Scientists have sampled those geysers remotely and found organic molecules, silica particles, and hydrogen inside the plumes. The water is coming from a liquid ocean beneath the moon's frozen crust. That ocean sits in total darkness, under pressure, above a rocky seafloor. After the dark oxygen discovery, astrobiologists began asking a pointed question. If rocky seafloors in cold, dark, high-pressure oceans can naturally produce oxygen through mineral electrochemistry, could the rocky floors of Europa's or Enceladus's oceans be doing the same thing? There is no confirmed answer. This is an active area of research and theoretical modeling. Scientists are building models of what mineral conditions on those moon's seafloors might look like, and whether the electrochemical mechanism for dark oxygen production could operate there. NASA has taken the Pacific seafloor discovery seriously enough to begin incorporating it into planning for future missions to the outer solar system. Confirmed by public statements from NASA researchers in 2024 and 2025, the search for life beyond Earth just gained a new tool, a new set of conditions to look for, a new type of environment to consider habitable. And it came from a pile of lumpy rocks at the bottom of the Pacific that scientists spent 8 years assuming were irrelevant. What happens when those rocks get destroyed before we fully understand them is the question coming next. Europa is covered in scars. Look at it through a telescope and you see a cracked, icy shell streaked with brown and orange lines cutting across a pale white surface. Those lines are fractures. The ice is moving. Something underneath is pushing it, shifting it, cracking it apart and refreezing it in new patterns over millions of years. That something is an ocean. Europa is slightly smaller than Earth's moon. It orbits Jupiter at a distance that puts it well outside the zone where sunlight could warm a planet enough for liquid water. By the basic rules scientists used for most of the 20th century, Europa should be a frozen dead rock, cold all the way through, nothing moving, nothing alive. But Jupiter is massive. Its gravitational pull on Europa is immense and uneven. As Europa orbits, Jupiter's gravity squeezes and stretches the moon in a constant cycle. That squeezing generates heat inside Europa's interior through friction, the same way bending a metal rod back and forth makes it warm to the touch. Scientists call this tidal flexing, and it pumps enough heat into Europa to keep a layer of liquid water beneath the ice from freezing solid. The ocean underneath Europa's ice shell is estimated to hold more liquid water than all of Earth's oceans combined. Confirmed by magnetic field measurements and gravitational data from NASA's Galileo spacecraft, which orbited Jupiter in the 1990s, that ocean sits in permanent darkness. No sunlight reaches it. The ice above is miles thick. The floor of the ocean is rocky, sitting on Europa's silicate interior. The pressure at the bottom of that ocean would be extreme by any measure. Before July 2024, most scientists modeling Europa's habitability focused on hydrothermal vents as the most likely energy source for potential life. Warm water rising from the rocky floor, driven by tidal heating from below, carrying minerals and chemical energy upward. After July 2024, the mineral electrochemistry of the Pacific seafloor entered the conversation. If the rocky floor of Europa's ocean contains minerals with the right electrochemical properties, the same basic mechanism that produces dark oxygen in the Clarion-Clipperton zone could theoretically operate there. Cold water, rocky floor, conductive minerals, pressure. Whether Europa's seafloor rocks carry the specific mineral composition needed to generate sufficient voltage for electrolysis is unknown. This is a theoretical area being actively modeled. Scientists cannot yet sample Europa's seafloor directly, but NASA's Europa Clipper spacecraft launched in October 2024 and is currently traveling toward Jupiter. Its instruments are designed to study the composition of Europa's surface, the thickness of its ice shell, and the properties of its ocean. The data it returns will help scientists evaluate whether dark oxygen-style chemistry is plausible on the moon's floor. The mission will take years to return its most detailed findings, but the Pacific discovery gave scientists a new question to ask of that data when it arrives. Something on the rocky floor of the Pacific has been making oxygen in the dark for millions of years. Something on the rocky floor of Europa's ocean might be doing the same. And if it is, the implications for what might be swimming through that dark water are difficult to overstate. Enceladus does something no other moon in the solar system has ever been caught doing. It shoots its own ocean into space. At the south pole of this small icy moon of Saturn, a series of cracks in the ice called tiger stripes blast columns of water vapor, ice particles, and chemicals hundreds of miles above the surface. The plumes are visible. They have been photographed. They spread out into Saturn's rings, actually feeding one of the planet's outer ring structures with fresh ice particles every second. NASA's Cassini spacecraft flew through those plumes multiple times between 2005 and 2017. The data it collected from those passes changed astrobiology permanently. Inside the plumes, Cassini detected water. Expected, but also organic molecules, the carbon-based chemical building blocks that life on Earth depends on, molecular hydrogen, which indicates that water is reacting with hot rocks somewhere below, silica nanoparticles, which form only when water sits in contact with rock at temperatures above 90 degrees Celsius, and phosphorus compounds confirmed in a 2023 analysis of the Cassini data, which represent one of the essential chemical ingredients for life as we understand it. Every single ingredient on the standard checklist for a habitable environment showed up inside those plumes. Underneath Enceladus's frozen shell, a liquid ocean sits in contact with a rocky sea floor. The tidal heating from Saturn's gravity keeps the water liquid in the same way Jupiter keeps Europa's ocean from freezing. The ocean is in total darkness, the pressure is high, the water is in long-term contact with the rocks below. Before the dark oxygen discovery, scientists focused almost entirely on the hydrothermal activity detected through the silica and hydrogen data. Hot vents on the seafloor of Enceladus seemed like the most plausible energy source for any potential chemistry of life. After the Pacific discovery, a second mechanism entered the model. The rocky seafloor of Enceladus may contain mineral deposits with electrochemical properties. If those minerals accumulate in conditions similar to the Pacific nodule fields, voltage differences could develop between different mineral layers. Clustered deposits could potentially reach the threshold for water electrolysis. Oxygen produced that way in Enceladus's ocean would dissolve into the water rather than escaping to space. Over geological time, the local concentration could build up enough to matter for chemistry. This is theoretical. No data currently confirms nodule-like formations on Enceladus's floor. The mission that could investigate this directly does not yet exist. But here is what scientists now know with certainty. Enceladus has water, organics, heat, minerals, and phosphorus. The only item on the checklist that remained uncertain before 2024 was a stable oxygen source in the dark environment of the seafloor. The Pacific discovery added a plausible natural mechanism for producing that oxygen without sunlight. Enceladus is now considered by many astrobiologists to be one of the most compelling places in the entire solar system to look for life. The plumes give scientists a way to sample its ocean without landing. Future missions could fly through those plumes with more sensitive instruments than Cassini carried. If something is alive in that ocean, the evidence might already be shooting into space, waiting for us to catch it. After the July 2024 publication in Nature Geoscience, NASA scientists at the Jet Propulsion Laboratory in California started paying close attention to the Pacific seafloor in a way they had not before. The reason was practical. If you want to design a mission that can detect dark oxygen production on the floor of an alien ocean, you need to understand exactly what that process looks like in physical and chemical detail. You need to know what instruments can measure it. You need to know what signatures it leaves in the water. You need to know what conditions it requires. The only confirmed example of dark oxygen production in a real ocean environment is the Clarion-Clipperton Zone, which makes the Pacific seafloor suddenly a testing ground for alien ocean science. Researchers from astrobiology programs began collaborating with the oceanographers who had studied the nodules. They wanted to understand the voltage profiles of nodule clusters in detail. They wanted to map the oxygen concentration gradients in the water directly above clusters. They wanted to identify chemical signatures in the water that might be detectable from a spacecraft flying through a plume or orbiting a moon. This is confirmed. Collaborative work between ocean scientists and astrobiologists was underway by late 2024, with at least 2 institutions publishing on the cross-disciplinary implications of the dark oxygen finding for missions to Europa and Enceladus. The practical challenge for any future mission to Europa or Enceladus is immense. Europa's ice shell is estimated to be between 10 and 30 miles thick. Drilling through it is beyond current engineering capability. The Europa Clipper mission currently traveling to Jupiter will study the Moon's properties from orbit, not from within the ocean. Enceladus offers a different opportunity. The plumes at its south pole are a direct window into the ocean below. A future mission flying through those plumes with instruments sensitive enough to detect dissolved oxygen, electrochemical signatures, or specific mineral compounds could potentially identify dark oxygen-style chemistry without ever touching the seafloor. The design of those instruments depends on knowing exactly what to look for. That knowledge comes from studying the Pacific. Scientists in 2026 are using autonomous underwater robots called ocean gliders to map the Clarion-Clipperton Zone in unprecedented detail. These robots drift through the water column at various depths, collecting continuous data on oxygen concentration, voltage gradients, mineral particle density, and biological activity. The mission, which launched earlier this year, is partly scientific and partly a proof of concept for the kind of autonomous sensing technology that a future alien ocean mission would need. The Pacific seafloor became, almost overnight, a laboratory for questions that reach billions of miles beyond it. And while scientists plan those future missions, a very different kind of planning is happening, much closer to home. 17 companies are preparing to send machines, not instruments, to the same seafloor. And their goal is not to collect data. Their goal is to collect the nodules. The contracts existed long before anyone knew about dark oxygen, starting in the 1970s, as deep-sea surveys began mapping the full extent of the polymetallic nodule fields in the Pacific. The commercial potential of the Clarion-Clipperton Zone became clear to mining companies and governments alike. The metals sitting in those nodules, cobalt, nickel, manganese, and copper, were already valuable then. Their value has only increased. An international body called the International Seabed Authority, created under the United Nations, oversees commercial access to the deep sea floor in international waters. The Clarion-Clipperton Zone lies in international waters. Under the framework established by the International Seabed Authority, contractors can apply for exploration licenses that give them the right to survey specific sections of the zone and prepare for extraction. As of 2024, 17 contractors held active exploration licenses for sections of the Clarion-Clipperton Zone. These contractors include companies backed by governments of Japan, China, South Korea, Germany, France, and several other nations, as well as private commercial ventures. The scale of what they are planning is significant. A commercial nodule harvesting operation would deploy large robotic collector vehicles to the seafloor. These machines would crawl across the mud, vacuuming up nodules and the top layer of sediment around them. The collected material would be pumped up through a riser pipe to a processing ship on the surface. The sediment, separated from the nodules, would be pumped back into the water column, forming a plume of suspended particles that would drift with ocean currents. The nodule recovery itself would be complete in each harvested area within the operational timeline of the mining campaign. The sediment plume generated could travel hundreds of miles from the collection site. A moratorium on commercial extraction has been debated but has not been globally enacted as of early 2026. Some countries and environmental organizations have called for a pause on mining until the ecological and chemical impacts are better understood. Others argue that the demand for the metals is urgent enough to proceed. The dark oxygen discovery arrived in the middle of an already contentious debate and sharpened it considerably. Before July 2024, the argument against mining focused primarily on the destruction of fragile ecosystems and the loss of biodiversity. Those arguments were real, but had limited traction against the economic case for extraction. After July 2024, a new element entered the argument. The nodules being targeted for removal are the same structures producing oxygen in the deep ocean. They are the same structures supporting ecosystems that took millions of years to develop. They are the same structures astrobiologists are now using as a model for alien ocean habitability. The debate over whether to mine them became significantly more complicated, and one specific tension at the center of that debate is almost painfully ironic. Picture a forest, old growth, hundreds of years in the making, every tree interconnected through root systems, fungal networks, and shared water sources. The birds, insects, mammals, and plants, all depending on each other in ways that took centuries to develop. Now picture removing the trees. The ecosystem does not pause. It does not adapt quickly. It collapses. And the recovery time, if recovery happens at all, is measured in generations. The Clarion-Clipperton Zone seafloor is that forest, except the trees took tens of millions of years to grow, and the recovery time, if the nodules were removed, would not be measured in generations. It would be measured in geological epochs. Polymetallic nodules grow at 1 to 2 millimeters per million years. a harvesting operation can remove them in months. The nodules that took 50 million years to form could be gone from a given section of seafloor within a single commercial campaign. On any human timescale, they would never come back. The immediate consequences for the ecosystem are layered. Sponges that colonize nodule surfaces would lose their substrate and die. The Casper octopus and other species that depend on nodule-attached sponges for reproduction would lose their nesting sites. The thousands of species found in nodule-rich areas, many still unnamed and unstudied, would lose their habitat structure. The sediment plume generated by the mining operation adds another layer of damage. Fine particles suspended in the water column settle over surrounding areas for months, potentially smothering organisms in adjacent zones that were not directly mined. The plume can travel far beyond the collection site before dispersing. Dark oxygen production in the mined area would stop. The nodule clusters that combined voltage to split seawater would no longer be there. The local oxygen supply they provided to the surrounding water would disappear. Species that depended on that oxygen, particularly any aerobic organisms adapted to a slightly elevated local oxygen level, would face conditions they had not experienced in the millions of years their lineage had lived in that zone. Whether the global ocean oxygen supply would be affected by mining in the Clarion-Clipperton Zone is unknown. The total contribution of dark oxygen to overall ocean oxygen chemistry has not yet been quantified. This is one of the central open questions in the field. Scientists do not currently have enough data to say whether removing the nodules from this one zone would have measurable effects on oxygen concentrations outside the immediate area. What is certain is the local effect. The ecosystem built around the nodules in any mined section would be effectively destroyed on any human timescale. Environmental groups, some governments, and a growing number of scientists are calling for the International Seabed Authority to implement a precautionary pause on mining until the full ecological and chemical roles of the nodules are understood. Whether that pause happens before the first commercial operation begins is a question with a ticking clock. And underneath it all is an irony that deserves its own chapter. The metals inside the polymetallic nodules are not just valuable, they are specifically the metals that the clean energy transition needs most urgently. Cobalt, nickel, manganese. These three form the core chemistry of lithium-ion battery technology, the same technology powering electric vehicles, grid-scale energy storage, and billions of consumer electronics devices worldwide. The global push to replace fossil fuel vehicles with electric ones has created an accelerating demand for these metals that current land-based mining operations are struggling to keep up with. Battery manufacturers are competing for supply. Governments are treating access to these metals as a national security issue. Supply chains are being mapped, contested, and restructured at a geopolitical level. Against that backdrop, the Clarion-Clipperton Zone looks like a solution. The nodules contain battery-grade concentrations of exactly the metals the clean energy sector needs. The deposit is enormous. The metals are sitting there in international waters, accessible in principle to any contractor with the right license and the right equipment. The argument for mining is direct. The world needs electric vehicles to reduce carbon emissions. Electric vehicles need batteries. Batteries need cobalt, nickel, and manganese. The seafloor has them in abundance. The argument against mining is equally direct. The nodules producing those metals are simultaneously producing oxygen, supporting ecosystems, providing habitat for thousands of species, and potentially serving as a model for understanding life on other worlds. Removing them to power the green energy transition would cause irreversible damage to a deep ocean system that took tens of millions of years to develop. The collision between these two arguments is genuine and unresolved. There is no clean solution currently available. Land-based mining for the same metals carries its own serious environmental costs. Mines in the Democratic Republic of Congo, which produces the majority of the world's cobalt, have been linked to severe ecological damage, child labor, and the displacement of local communities. The choice between seafloor mining and land mining is a choice between two forms of significant environmental harm. Battery technology is evolving. Some manufacturers are actively working to reduce or eliminate cobalt from their battery chemistry. Lithium iron phosphate batteries, which use no cobalt at all, are gaining market share. But they have lower energy density than cobalt-containing batteries, which limits their use in high-performance applications. The timeline matters. If battery chemistry shifts away from cobalt and manganese before large-scale seafloor mining begins, the economic case for the Clarion-Clipperton Zone collapses. If mining begins before that shift happens, the damage to the nodule ecosystem will be locked in permanently. Scientists, engineers, policymakers, and companies are moving on different timescales toward different goals. The seafloor is sitting in the middle of all of it, producing oxygen quietly in the dark, unaware that a decision made in a boardroom or a government office could end that process within years. And the species living in that dark world, the ones that have never seen a sun and never will, have no voice in any of it. Pull the battery out of an electric vehicle and look at what is inside. The chemistry varies slightly by manufacturer, but the core materials are consistent. Lithium moves the charge, cobalt stabilizes the battery, nickel boosts energy density, manganese adds structural stability to the electrode. Those four elements, working together in precise proportions, allow a battery to store enough energy to push a 2-ton vehicle down a highway at freeway speeds for 300 miles on a single charge. Without cobalt, the battery degrades faster. Without nickel, the energy density drops. Without manganese, the electrode becomes unstable under repeated charge cycles. Every major electric vehicle maker on Earth is managing the supply of these metals as a core business problem, not a peripheral concern. Now consider the scale of what is happening globally. In 2024, electric vehicles represented roughly 20% of all new car sales worldwide. Projections suggest that number rises sharply through the end of the decade. Each electric vehicle contains roughly 10 to 20 pounds of cobalt in its battery pack, depending on the chemistry used. Scaling that across hundreds of millions of vehicles represents a cobalt demand that current landmines cannot easily meet. Nickel and manganese face similar pressures. Energy storage for solar and wind power also uses large battery systems built from the same materials. Grid-scale storage, the technology that stores renewable electricity for use when the sun is not shining and the wind is not blowing, requires enormous quantities of exactly the metals sitting in the nodules. The Clarion-Clipperton Zone is estimated to hold more nickel, cobalt, and manganese than all currently known land reserves combined. That number is confirmed by geological surveys, but should be understood in context. Surveying the ocean floor is difficult, and exact reserve estimates carry significant uncertainty. What is certain is that the metals are there in high concentrations across an area the size of the continental United States. The pressure on policymakers to allow extraction is real. Countries that control access to battery metals gain significant leverage in the global economy. The green energy transition, which most governments have committed to pursuing, requires those metals on a timeline that feels urgent. The Pacific discovery did not slow that pressure. In some ways, it intensified the conflict by making the choice more visible. Extracting the nodules is no longer simply a question of ecological damage to some poorly understood deep-sea habitat. It is a question of whether to destroy a confirmed oxygen-producing system, a confirmed biodiversity hotspot, and a confirmed scientific resource in order to accelerate a transition that, by its own logic, is supposed to protect the planet. The contradiction is uncomfortable, and the companies holding the exploration licenses are aware of it. What they have proposed as a solution is something worth examining very carefully. Scientists have named roughly 5,000 species from the Clarion-Clipperton Zone. That sounds like a large number. It is a fraction of what lives there. The zone covers an area the size of the continental United States. The biological surveys conducted so far have sampled a tiny percentage of that total area. Researchers pull a collecting device across the seafloor gather what they can from a small patch of mud, and bring it to the surface for study. The process is slow, expensive, and limited by the reach of the equipment. From those samples, scientists consistently find organisms they have never seen before. New species appear in nearly every collection. Worms with body structures unlike any documented relative, mollusks adapted to conditions so specific that they exist in only one type of microhabitat, microbial communities embedded in the sediment that respond to the chemical environment created by the nodules. The species total for the full Clarion-Clipperton zone, including microbes and everything too small or rare to have been collected yet, is estimated by some researchers to be in the tens of thousands. Most of those species have never been photographed. Many have never been collected. A significant number have probably never been observed by any human instrument at any point in history. Each of these species is the product of millions of years of evolution under very specific conditions. The pressure, the cold, the dark, the chemistry around the nodules, the local oxygen levels maintained by the dark oxygen process. Every organism living in that zone has adapted to exactly the environment it is in, and that environment includes the nodules as a structural, chemical, and oxygen-producing component. Remove the nodules, and the environment changes immediately. The hard surfaces disappear, the local oxygen chemistry shifts, the mineral gradients around cluster sites flatten out, the microhabitats dissolve. Organisms adapted to those specific microhabitats have nowhere to go. The flat mud surrounding the nodule fields does not offer the same conditions. The next nodule field in the Pacific may be hundreds of miles away, separated by stretches of seafloor with completely different chemistry. Deep sea organisms do not migrate the way surface animals do. They are not built for movement across vast distances. They are built to stay exactly where they are in the conditions they evolved for. Extinction of species that have never been formally identified is not a hypothetical concern in conservation biology. It has happened before in other environments. Forests cleared before the species inside them could be studied. Reefs bleached before the organisms living in them could be catalogued. The loss is permanent and invisible, recorded nowhere because the observation never happened. In the Clarion-Clipperton zone, the observation is barely beginning. Scientists are in the earliest stages of understanding what lives down there. The discovery of dark oxygen itself came only in 2024, after 8 years of instrument recalibration. What other discoveries are waiting inside that mud? That question has no answer yet, and mining could close it permanently before anyone gets the chance to ask it properly. The deep sea beyond national waters belongs in principle to everyone. That principle is enshrined in the United Nations Convention on the Law of the Sea, a treaty signed in 1982 that established the legal framework for international ocean governance. Under that framework, the seabed in international waters is designated as the common heritage of mankind, a legal concept meaning that no nation owns it and any resource extraction must be managed collectively for the benefit of all people, including future generations. The International Seabed Authority, based in Kingston, Jamaica, was created to implement that principle. Member states of the United Nations give the authority the power to approve exploration contracts, set environmental standards, and eventually approve commercial mining operations in international waters. The authority has 36 members on its governing council and a full assembly of all member states. Decisions about mining regulations require negotiation among nations with very different interests. Countries with significant mining industries see the nodule fields as an economic opportunity. Countries with smaller economies and limited industrial capacity see them as a shared global resource that should generate revenue for development. Environmental advocates within member states push for stronger protections. The process of establishing a commercial mining code, the full set of rules governing how nodule extraction must be conducted, has been ongoing for years. As of early 2026, a final commercial mining code has not been formally adopted by the authority. The absence of that code creates a legal grey area. One nation, Nauru, a small Pacific island state, used a provision in the treaty to trigger a deadline in 2021 that required the authority to either finalize the mining code or allow applications to proceed under existing rules. That trigger set off renewed urgency in negotiations that continue today. Some member states, including France, Germany, and Chile, have called for a precautionary pause on mining until environmental standards are finalized and the ecological role of the nodules is better understood. The dark oxygen discovery was cited explicitly in some of those calls. Other member states and contractors argue that the legal framework already provides sufficient protection and that further delays cost economically significant opportunities. No single country, organization, or court currently has the power to unilaterally stop a contractor that has received approval from the International Seabed Authority from proceeding. The legal structure distributes authority across member states, and consensus on a moratorium has not emerged. The outcome of this governance question will determine whether the Clarion-Clipperton Zone is mined in the coming years, or whether a period of scientific study and environmental assessment precedes any extraction. The seafloor does not care about the legal debate. The nodules keep generating voltage, the oxygen keeps rising, and above them, a new expedition arrived in 2026, carrying tools that could finally measure the full scale of what the nodules are doing. Scientific papers usually reach a small audience. Researchers in the relevant field read them. Some journalists who cover science report on them. The wider public rarely notices. The July 2024 publication in Nature Geoscience was different. Within 48 hours of publication, the paper had been covered by major news outlets across 6 continents. The story of oxygen being produced in total darkness at the bottom of the Pacific Ocean was picked up by general audiences in a way that most oceanography research never achieves. Social media amplified it. Science communicators translated it into accessible language within hours. The phrase dark oxygen became briefly but genuinely famous. The scientific reaction was intense. Researchers in marine biology, geochemistry, astrobiology, origin of life studies, and deep-sea ecology all had immediate responses. Some were enthusiastic, others were skeptical. Several research groups publicly questioned specific aspects of the methodology and called for independent replication. The skeptical responses focused on a few key points. Some researchers questioned whether the pressure chamber experiment fully replicated deep-sea conditions. Others pointed out that the oxygen measurements in the field, while confirmed by 2 different instruments, still relied on indirect evidence for the nodule mechanism specifically, proving that the nodules were the source, rather than some other process happening near the seafloor, required ruling out alternatives more completely than the 2024 paper had done. Andrea Sweetman's team acknowledged the critiques and invited independent research. By the end of 2024, at least 3 separate research groups had initiated their own field studies in the Clarion-Clipperton Zone to attempt replication. The debate within science over the paper's conclusions is ongoing and healthy. This is how science works. A finding is published. The community tests it. If it holds up under scrutiny from multiple independent groups, it becomes established knowledge. If it does not, it is revised or discarded. The mechanism of dark oxygen production, electrolysis driven by nodule voltage, is the specific claim under examination. The existence of anomalous oxygen in the deep seafloor environment was already accepted before the paper. The nodule mechanism as the explanation is what the scientific community is now stress testing. What the paper unambiguously achieved, regardless of how the scientific debate resolves, is a permanent shift in the questions scientists are asking. Before July 2024, the possibility of oxygen production in deep, dark, cold water by purely mineral means was not a subject of active research. After July 2024, it became one. New expeditions were funded, new instruments were designed, new collaborations formed across disciplines that rarely talk to each other. Astrobiologists and oceanographers started meeting regularly for the first time. The paper may be revised, the mechanism may be refined, Some of the details may change as more data comes in. The question it opened will not close. And in 2026, a new set of machines went back down to find the answers that the 2024 paper left unresolved. Earlier this year, a fleet of autonomous underwater robots descended into the Pacific Ocean above the Clarion-Clipperton Zone. These machines are called ocean gliders. They're roughly the size and shape of a small torpedo, about 6 feet long, with fixed wings and no propeller. They move through the water by changing their buoyancy, inflating and deflating an internal bladder to sink or rise, and using their wings to convert that vertical movement into forward gliding motion. They use almost no power. A single battery charge can keep them operating for weeks or months. Ocean gliders are not new technology. They have been used in oceanography for decades. What is new is the specific mission these gliders are running in 2026. Each glider in the current expedition carries a set of sensors designed specifically around the dark oxygen question. Standard oxygen sensors measure dissolved oxygen in the water column as the glider moves through it. Additional sensors measure electrical conductivity gradients in the water, looking for the signature of voltages moving through seawater from nodule clusters below. Chemical sensors detect specific mineral compounds dissolved in the water that would indicate active electrochemical reactions at the seafloor. The gliders are mapping the water column at multiple depths, from just above the seafloor up through several hundred feet, recording continuous data as they traverse the zone. The goal is to build a three-dimensional picture of where oxygen anomalies occur, how strong they are, how they relate to the distribution of nodule clusters below, and how they vary across different sections of the Clarion-Clipperton Zone. This dataset, when complete, will give scientists their first comprehensive estimate of total dark oxygen production across the zone. It will also reveal whether production is continuous or intermittent, whether it varies seasonally, and whether some areas are significantly more active than others. The expedition is a collaboration between several universities and oceanographic institutes across the United States, Germany, and Japan. Funding came partly from ocean research agencies and partly from a specific allocation tied to evaluating the environmental impact of potential mining operations. That second source of funding reflects the direct connection between the scientific question and the policy debate. Regulators deciding whether to approve commercial mining licenses need to know the ecological function of the nodules before granting removal rights. The 2026 glider expedition is partly designed to generate that information. The results are expected to be published in stages through late 2026 and into 2027. The first data releases have already begun to sketch out a picture of the zone's oxygen dynamics that is more complex than anyone expected. Some areas show strong oxygen anomalies in the water column above dense nodule clusters. Others show almost none, even where nodules are present. The reasons for that variation are not yet understood. The answer to that variation might be the most important piece of the puzzle still missing. The Clarion-Clipperton Zone is one nodule field, a large one, but one of many. Polymetallic nodule deposits have been found in multiple ocean basins: the Peru Basin in the South Pacific, the Central Indian Ocean Basin, the South Atlantic, the Cook Islands region. Each of these areas holds nodule fields with similar mineral compositions to the Clarion-Clipperton Zone, formed through the same geological processes over similar timescales. If the dark oxygen mechanism operates wherever the right mineral conditions exist, the global scale of the process could be substantially larger than any single field suggests. Scientists are now attempting to evaluate whether anomalous oxygen readings appear in the deep water above other nodule fields around the world. This is a coordinated international effort that grew directly out of the 2024 publication and the global attention it generated. The challenge is the same one Sweetman's team faced in 2013. Deep ocean oxygen measurement at extreme depths is expensive, slow, and technically demanding. Deploying sensors to 13,000 feet in a remote patch of the South Atlantic or the Central Indian Ocean requires a research vessel, specialized equipment, and significant institutional support. The number of research institutions capable of running those expeditions is limited. The number currently funded to do so is smaller still. As of early 2026, preliminary surveys have been conducted in the Peru Basin and one area of the Central Indian Ocean. Early results from both locations show elevated oxygen in the water column above nodule-dense regions, compared to adjacent areas without significant nodule coverage. These findings are preliminary, have not yet been peer-reviewed, and should be treated as early signals rather than confirmed results. If the preliminary results hold up under full analysis, the global ocean picture becomes significantly more complex. Dark oxygen production would represent a distributed geological process operating across multiple ocean basins simultaneously, quietly feeding dissolved oxygen into deep water that scientists had assumed was purely a zone of oxygen consumption. The implications for ocean chemistry models would be substantial. Current models of how oxygen moves through the global ocean are built on the assumption that all oxygen enters from the surface, either dissolved from the atmosphere or produced by surface photosynthesis. If a second source is operating at depth across multiple basins, those models need to be revised. Revised models would change estimates of how much oxygen the deep ocean can support, which species distributions are sustainable in which areas, and how the deep ocean will respond to changing conditions as the climate continues to shift. Scientists do not yet know how significant dark oxygen is in the global oxygen budget. That answer is one of the central unknowns the current wave of research is trying to resolve. What they do know is that a process they did not account for in any of their models has been running below their instruments the entire time. And it has been running for far longer than anyone initially guessed. Everything confirmed so far about dark oxygen describes a snapshot. Sweetman's team measured rising oxygen in the Clarion-Clipperton zone at specific moments over a 10-year period. The laboratory pressure chamber confirmed the production mechanism under controlled conditions. The 2026 Glider expedition is mapping the current distribution of oxygen anomalies across the zone. All of that data captures what is happening now or over the past decade. None of it tells scientists whether dark oxygen production runs at a constant rate through geological time, or whether it pulses in cycles tied to conditions that change over longer periods. This distinction matters enormously. If dark oxygen production is continuous and stable, then the total oxygen contributed to the deep ocean over millions of years is enormous. Species evolution, ocean chemistry, and potentially the history of atmospheric oxygen on Earth all need to account for a steady, geological-scale source of oxygen that was previously invisible to science. If dark oxygen production is intermittent, cycling on and off in response to changing ocean chemistry, geological activity, or nodule formation rates, then its cumulative contribution depends entirely on the timing and duration of those cycles. The evolutionary and chemical implications could be much smaller, or they could be concentrated in specific geological periods when conditions were right. The nodules themselves offer one way to investigate this question. Because each layer of a nodule records the chemical conditions present when that layer was deposited, researchers can look for signatures of active electrolysis at different depths within a nodule structure. If specific chemical markers of water splitting appear consistently across many millions of years of nodule layers, that suggests continuous production. If the markers appear only in certain layers, that suggests cycles. This type of analysis is confirmed to be underway in several research laboratories. Results have not yet been published. A second approach involves examining the sediment surrounding nodule clusters for chemical signatures of long-term oxygen exposure. Sediment records stretch back millions of years and can be sampled with long-core drills. If the sediment chemistry directly around nodule clusters differs from sediment further away in a pattern consistent with decades or centuries of elevated local oxygen, that provides indirect evidence for sustained production over geological time. These investigations are in early stages. No peer-reviewed conclusions have been published on the continuity question. What scientists agree on is that the question is not trivial. The answer will determine how significantly dark oxygen needs to be incorporated into models of Earth's oxygen history, deep ocean ecology, and the search for life beyond Earth. An intermittent process matters. A continuous one changes the textbooks more fundamentally. Right now, nobody knows which one it is, but the next piece of this story goes further back in time than any instrument can measure directly, and asks a question that changes the meaning of every breath you have ever taken. The oxygen in the air you are breathing right now has a history. Every oxygen molecule in Earth's atmosphere was produced at some point in the past by a chemical reaction that split it away from another molecule. For most of Earth's history, that reaction was photosynthesis. Cyanobacteria, the ancient single-celled organisms that first evolved the ability to use sunlight to split water, began pumping oxygen into the ocean and atmosphere roughly 2.4 billion years ago in an event called the Great Oxidation Event. Before the Great Oxidation Event, Earth's atmosphere contained almost no free oxygen. After it, oxygen levels rose over hundreds of millions of years to something approaching what we have today. Every animal that has ever breathed oxygen, every aerobic organism that has ever lived, owes its existence to what those ancient cyanobacteria started. That story is confirmed and well established. The question the dark oxygen discovery raises is whether it was the only story. If polymetallic nodule fields have existed on the deep sea floor for billions of years, and if the electrochemical mechanism for dark oxygen production has been running throughout that time, then a second source of oxygen has been feeding the ocean for far longer than the Great Oxidation Event. A slower source, a quieter one that operated in total darkness, miles below the surface, adding dissolved oxygen to the deep ocean independent of anything happening at the surface. Whether that process is old enough and large enough to have meaningfully influenced Earth's atmospheric oxygen history is unknown. Confirmed as unknown. The geological record for dark oxygen production over billions of years does not yet exist. The tools for reading that record in nodule layers and deep sediment cores are only now being designed. Some scientists believe the contribution of dark oxygen to the pre-Great Oxidation atmosphere may have been small relative to what photosynthesis eventually achieved. Others argue that even a small persistent source of deep ocean oxygen, operating for billions of years before photosynthesis evolved, could have played a role in making the ocean hospitable enough for the organisms that eventually evolved photosynthesis. A chicken and egg problem sits at the center of this question. Did photosynthesis produce the oxygen that allowed complex aerobic life to evolve? Or did a pre-existing baseline of dark oxygen help create the conditions in which photosynthetic organisms could first emerge? Science cannot answer this yet. The evidence needed to resolve it requires reading chemical signatures in geological formations that are billions of years old, using methods still under development. What the dark oxygen discovery confirmed is that the question is no longer absurd to ask. Before July 2024, proposing that the seafloor had been producing oxygen independently of sunlight for billions of years would have required extraordinary evidence to even be taken seriously in a peer review context. After July 2024, it is a legitimate open scientific question, and the most personal version of that question is the one that comes next. Every major story about life on Earth begins near the surface. The first photosynthetic cells floating in sunlit shallow water, the first land plants pushing up through soil toward the sky, the first lungs breathing air at the edge of an ancient shoreline. Every chapter in the history of complex life points upward, toward light, toward the surface, toward the sun as the engine of everything. The dark oxygen discovery asks a different question. What if the very first aerobic chemistry on Earth did not start at the surface? What if the conditions for oxygen-based life first appeared in the dark, miles below, in the mineral fields of an ancient seafloor? Here is the sequence that some researchers now find worth taking seriously. In the early history of Earth, before photosynthesis evolved, the ocean covered most of the planet's surface. The seafloor was rocky. Mineral deposits formed through the same slow geological processes that still operate today. If nodule-like formations developed on that ancient seafloor, and if they carried electrochemical properties similar to modern nodules, then electrolysis-driven oxygen production may have been running in the deep ocean long before any living organism learned to produce oxygen through photosynthesis. That dark oxygen, dissolving slowly into deep water, may have created a chemical environment in the abyss where molecules capable of using oxygen in their reactions had an advantage. Simple chemical reactions that run on oxygen could accumulate, concentrate, and eventually become organized enough to be considered proto-life. By the time the first photosynthetic organisms evolved near the surface, the deep ocean may have already been a working laboratory of aerobic chemistry for an incomprehensible span of time. This is a theory. It is speculative, supported by the confirmed mechanism of dark oxygen production, and by theoretical modeling of early Earth chemistry, but far from proven. The evidence needed to test it directly may not be recoverable from the geological record. What gives it weight is the nature of the nodule chemistry. The ingredients are specific, minerals with electrochemical properties, conductive seawater, the exact voltage threshold for water electrolysis. These are not vague conditions. They are precise, measurable, and real. They exist on the modern seafloor. Whether they existed on the early Earth seafloor is a geological question with a plausible yes answer, because the mineral processes that create nodules are driven by chemistry that operated on early Earth as well. The rock at the bottom of the Pacific may be the closest living relative of a process that breathed its first oxygen into the ancient ocean, long before any lung existed to receive it. And the question that follows from that is one scientists are now brave enough to say out loud: alive is a word with a definition. In biology, something is considered alive if it metabolizes energy, responds to its environment, and reproduces. Rocks do not metabolize, rocks do not reproduce. By every standard definition, the polymetallic nodules on the seafloor of the Pacific are not alive. And yet, they grow, slowly, over millions of years, adding layer by layer in response to the chemical composition of the water around them. They generate electrical charge. They react with their environment in a way that splits water molecules apart and releases oxygen. They create conditions that allow other organisms to live, reproduce, and evolve. They change the chemistry of the ocean around them in ways that ripple outward through the ecosystem for miles in every direction. They do something remarkably similar to what living things do. They take in energy from their environment, process it, and release a product that other systems depend on. The energy source is electrochemical rather than metabolic. The timescale is geological rather than biological. But the functional relationship between the nodules and the ecosystem around them looks in important ways like the relationship between a living organism and its environment. Some researchers in the field of geobiology, the study of how geological and biological processes interact, are beginning to ask whether the rigid boundary between living and non-living is as clean as biology has assumed. The nodule fields complicate that boundary in a way that is difficult to dismiss. The seafloor is doing something sustained, something consequential, something that shapes the conditions for life across thousands of miles of deep ocean. Whether that something constitutes a form of aliveness that science has not yet named, or whether it is simply chemistry that mimics aliveness closely enough to make us uncomfortable, is not a question that can be resolved with the tools currently available. It is a question that requires new frameworks, new language, new ways of thinking about what it means for a system to be alive or to behave like one. The nodules have been sitting in the dark for tens of millions of years, doing what they do, without any observer to notice. Sweetman noticed in 2013. The world found out in 2024. It has now been over a decade since a researcher stared at an impossible number on a sensor screen and started asking what it meant. The answer has expanded outward from that moment in ways that touch the origin of life, the search for alien worlds, the future of the clean energy economy, the survival of species never photographed, and the deepest question in biology. And the rocks on the seafloor keep generating voltage. The water keeps splitting. The oxygen keeps rising through 13,000 feet of dark water toward a surface that knows nothing about where it came from. Something extraordinary is happening at the bottom of the Pacific. We are only just beginning to understand what it means.